All right, hello everyone. We're going to start chapter 8, cellular respiration, and this is the chapter that follows photosynthesis. So we're going to be looking at how glucose, which was produced during photosynthesis, is going to be used within eukaryotic cells, specifically within the mitochondria, and what that energy or how that energy is going to be transferred to other molecules. So without further ado, let's get into it. The first section, just like in photosynthesis, just kind of gives you an overview of this kind of this process. This is just the process where we're going to break down glucose in the form of C6H12O6, and we're going to produce carbon dioxide, water, and energy. Now, we're not really producing the energy, we're just transferring it to other molecules. One other molecule that's going to be needed in this step is going to be oxygen. And if you notice here, these two inputs for cellular respiration or reactants are going to be the products of photosynthesis. So what photosynthesis gives us, we are going to take, break down, and use to form CO2 and water. Now, these two products are going to go back into photosynthesis and be combined together to form sugar and oxygen. So this is kind of like not a reverse reaction, but it's like almost like a coupled reaction where the products are the reactants of the next reaction. But the end goal here is to produce ATP, which is going to be our high energy molecule. And we produce around 36 to 38 of them for glucose, which is, we'll talk about later on why it's between these numbers and not a specific exact number. I love this image you can see here. You have food powering the production of ATP. That production of ATP then produces other cellular, or it powers other cellular just processes in general. Now, two molecules we have to know about before we get into this chapter are NADH and FADH2. They are reusable, and they are just like NADPH from the last section or last chapter. These molecules are going to specifically transfer electrons and hydrogen ions to the next couple steps. So these are just taxis for these two items. They're very, very close to the NADPH molecules we saw in the photosynthesis. The only difference is this one has an extra phosphate group. All right, cellular respiration. So we have 4 different steps here, and we should know the difference here. Aerobic requires oxygen. Anaerobic doesn't require oxygen. The only step that's anaerobic is glycolysis, and again, we'll get to more about why that is later on. But then we have the prep step, Krebs cycle, and electron transport chain. These 3 steps are aerobic. So if you don't have oxygen or you don't have enough oxygen, these 3 steps will not work. Glycolysis will occur whether there's oxygen or not. Barring that there are anaerobic processes occurring. But yes, glycolysis, anaerobic, the next 3 steps are aerobic. Now, we have to also kind of understand where parts of the mitochondria are. So these are the mitochondria. They are multiple per cell, has double membrane. Remember the endosymbiotic theory. Matrix, this is the fluid inside the mitochondria. So this is the fluid. This is kind of that light yellow portion here. Cristae, these are folds within the inner membrane. You can see we have 2 membranes, so these are the folds here. This increases surface area for more cellular respiration. We'll get into more surface area and volume ratios when we get to cells in mitosis, but intermembrane space, this is the location between the 2 membranes. And if you think about it, we're compartmentalizing here. So everything that's going on right here is compartmentalizing. We're making a larger area smaller and giving it specific jobs. So this is just kind of like the basis of what the mitochondria is, where locations are. You have to know where all these locations are: inner, outer membrane, intermembrane space, matrix, and these folds, which are called cristae. All right, this is where 8.1 stops. All right, glycolysis is the first step, and we're going to focus on it because it occurs first. Now, glycolysis, glyco means sugar, lysis means to break, so think breaking down sugar. This whole step breaks down sugar into 2 pyruvate molecules, and just like in photosynthesis, focus on the carbons. Now, this step occurs first because it evolved first. And it evolved first because it evolved when there was a time when there was no mitochondria or no other really major, I don't say major organelles, but the mitochondria just wasn't there. So it occurs in the cytoplasm because it evolved first. Then the other steps evolved in the mitochondria, which further breaks down those molecules. Now, there was no oxygen in the atmosphere at this time. So again, this one doesn't require oxygen. Now, there's a lot that's going on here. I'm going to go over just like the notes and come back to this, but this is basically what you have to know for glycolysis. So let's move forward real quick. There's two main steps here. We have the energy investment step and the energy harvesting step. In this first part, there are two ATP used that are going to be used up, and they're going to add their phosphate groups onto glucose. Now, glucose then splits into two G3P molecules, and you might remember those from photosynthesis. Yes, we saw those in photosynthesis. Remember, I told you they are very versatile molecules. So we're going to see them again here. So our first thing that happens is glucose is broken down to two G3P molecules, and we use two ATP for their phosphate groups. Now, the second step is going to be the energy harvesting step. Now, this occurs per pyruvate. G3P is oxidized into is oxidized by NAD plus, which becomes NADH. This just means, again, oxidation reduction reactions, oil rig. Oxidation is losing, reduction is gaining. So G3P is going to lose electrons and hydrogen ions, and the NAD is going to pick them up and take them to a later location. This also creates two ATP molecules per G3P, but we have to double everything because, again, we have two pyruvates here. Now, let me go back here. So again, we have our energy harvesting step. You can see two ATP are going to be used up. We phosphorylate glucose. It breaks down into two G3P molecules. Now, in all reality, it does break down to something called DHAP, but DHAP is converted then into G3P. So you don't have to know that second step, but just understand this produces two G3P molecules. Now, per each G3P, you're going to produce an NADH and two ATP, but remember, we have to double that because we get two G3Ps here. So at the end, we get two pyruvates, 4 ATP, but we only get a net of 2 ATP because we had to spend some ATP up here. I always like to remind my students, you have to spend money to make money. So we used up 2 ATP, we produced 4 ATP, but it's only a net gain of 2. We also produced 2 NADHs. So going on, one thing I want you to understand here that this phosphorylation, I can't get my slides here, is called substrate level phosphorylation. We're just taking an enzyme, or an enzyme is passing a phosphate group from one molecule to the next. This is just called substrate level because the substrate here is a molecule with a phosphate group. This is kind of easy to understand. The next type, which we'll get later on, is called oxidative. That's a little bit more difficult. So again, like I said, outputs 2 net ATP because we produced 4, but we had to use 2. We produced 2 NADHs, and we produced 2 pyruvate molecules, which each have 3 carbons. Yeah, this is how I kind of look like after explaining this up because there's so much going on here. But this is the end of 8.2. The next section is 8.3, which is going to be, let's see here, the prep step. Here we go. All right, 8.3 is going to be looking specifically at the prep step. Let me go back here. This is also known as pyruvate decarboxylation, and this is a big word, but if we break it down, pyruvate talks about the molecule that's going to be is going to be a reactant for this process, and decarboxylation. Carboxylation means we add a carbon. Decarboxylation means we're going to remove a carbon. And basically what happens here is the pyruvate is going to move from the cytoplasm into the mitochondrial matrix, and it does this by going through transport proteins through each membrane. Right here, the inner membrane and the outer membrane. So this reaction is going to occur in the mitochondrial matrix. What happens is pyruvate is going to be converted into an acetyl-CoA. Now, pyruvate has 3 carbons, and acetyl-CoA only has 2. So it means we're going to lose a carbon. We always lose carbon in these steps in the form of CO2. What's going to happen is we're also going to reduce an NAD into NADH. And what's going to happen is this coenzyme A is going to come in here. It's going to bind with the acetyl group. Now, coenzyme A is a really big molecule, and I love this meme. I don't know who made this meme. I wish I could say I made it, but I didn't. This acetyl-CoA is a huge molecule here. And what happens is the acetyl part's very small, but this coenzyme A is pretty big. And I love this video of SpongeBob. Oh my gosh, it's too good. This coenzyme A is this really big fish here. And then the little acetyl group's Patrick. Basically what this coenzyme A does is it's just like a transporter for acetyl-CoA, or for the acetyl group, and it helps it bind into the next step, which we're going to see here in a little bit. So again, during this process, NAD becomes NADH, CO2 is produced, and this occurs twice because we get 2 pyruvates in through this step here, and we are going to get both these pyruvates from the first step, and they're going to be both changed into acetyl-CoA, so we have to double everything for this step. But yeah, on to 8.4. All right, 8.4 is going to talk about the Krebs cycle. I call it the Krebs cycle, but it's also known as the TCA cycle or the citric acid cycle. It has 3 names. Again, we like to name things in science like multiple times just to confuse you. Not really. That's a joke, by the way. But this just happens. But basically, it was discovered by Hans Krebs here. And the joke is, yeah, there's many names for this, but I call it the Krebs cycle specifically. It occurs in the mitochondrial matrix. It's the the ATP that's produced here is produced by substrate level phosphorylation, and we'll get back to that when we look at the actual cycle. This occurs twice per glucose molecule, and that's because in the mitochondria, we are going to intake 2 pyruvates that get changed into 2 acetyl-CoA. So both these acetyl-CoAs will go into this cycle, and it will make it turn twice then. So this is the basic cycle. I'm just going to kind of review it just as the image here, because it's a little bit easier than to just write everything down. I'll show you what you have to write down here next. But basically, the acetyl-CoA comes in here, and the coenzyme A, the CoA part here, drops off the acetyl group, which binds with oxyl acetate, which is a 4-carbon molecule. This 4-carbon molecule plus this 2-carbon molecule creates this 6-carbon molecule, and it's called citrate. That's why they call it the citric acid cycle, because citrate and citric acid are the same thing. Again, we'll focus on the carbons. 2 carbons plus 4 carbons equals 6 carbons. What happens is you are going to go through a reaction where a carbon is released in the form of CO2. We always release carbon in the form of CO2. This accounts for the carbon that you breathe out. That carbon dioxide is what is from, part of it is from this step. We also change an NAD into an NADH. Remember, that is a reduction reaction because we're gaining an electron. Now, this 5-carbon molecule is called ketoglutarate. I specifically only really want you to remember the citrate molecule. That's it. But here's just for context what's ketoglutarate. It looks like it's a 5-carbon molecule. This reaction occurs again where you lose a carbon in the form of CO2. So again, this is what leaves our body through our lungs. We produce another NADH, and it becomes succinate, which is a 4-carbon molecule. This 4-carbon molecule gets changed into fumarate, and does this by changing FAD into FADH2. It reduces it, which means this molecule is oxidized. It says GDP here and GTP. This is another energy molecule. Just think of this as ATP. I don't get into the specifics here, but just remember this as ATP instead of GDP and GTP. So it makes an ATP molecule here. Fumarate is changed into oxyl acetate, if you notice here. And here we do not lose carbon. So in this case, we're not losing carbon. We're at the same number of carbons. So 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. We're also going to produce an NADH here as well. So this then turns twice because the acetyl-CoAs coming in from the last step make it turn twice. Here's what you have to write down. Now, I have you not memorize the actual names here. I have them in red. I still want you to write them down, but just understand that the names aren't that important. I want you to mainly know the acetyl-CoA and the citrate here. All right, moving on. So our products here, we produce 4 CO2, 2 ATP, 2 FADH2, and 6 NADH. And you might think, hey, why are we doubling everything? Remember, this turns twice, so we have to double everything for this process. All right, this is the end of 8.4. 8.5 is going to specifically look at the electron transport chain, and this is going to be one of the more difficult sections because it's just so complicated. But don't let that overwhelm you. I say that, but it's not as bad because we already learned about the electron transport chain in photosynthesis. So it's going to really mirror that process. So this occurs along the intermitochondrial membrane. It will still deal with the intermembrane space in the matrix, but more specifically, we're looking at this intermitochondrial membrane here. This is that intermembrane of the mitochondria. What happens is the NADH and FADH2 molecules are going to deliver their electrons and hydrogen ions to the matrix and to the intermembrane proteins. So you can see NADH oxidizing, FADH2 oxidizing, and dropping off those electrons to these electron transport chain proteins. What's going to happen is these electrons are going to flow through those proteins, and they're going to do the same exact thing that they did in the electron transport chain for photosynthesis. They are going to use that energy to force more hydrogen ions into this intermembrane space. So this intermembrane space is almost going to act like those thylakoids. Again, we're compartmentalizing. We're making a larger area smaller, and we're putting those protons into that space. Again, when we reduce that area, it's much easier for us to raise concentration rates. And when that happens, it is easier to create a concentration gradient, which we're going to use to produce ATP. Now, the electrons are going to move through the electron transport chain because each protein here is a little bit more electronegative, which means it wants to pull on these electrons. So as the electrons move from protein to protein, they're going to jump from protein to protein because each protein is a little bit more electronegative than the last one, which means they want to pull on. So this one's the least electronegative, this one's the most. It pulls them through this electron transport chain. Now, at the end there, you're going to see that the hydrogen ions and the electrons are going to bond with an oxygen molecule. This is going to be the final electron acceptor. And yes, this is pretty important. This oxygen molecule has to be there for the electrons to bind with, and this is going to form water. And if you remember back from photosynthesis, what was the first step of photosynthesis? Water breaking down, donating their electrons and hydrogen ions, and forming oxygen. This is the last step of cellular respiration. This is almost like a reverse reaction. It's not a perfect reverse reaction. Obviously, there's a lot more different molecules. But in this step here, this is like the exact opposite of what happens. We form water by using oxygen, hydrogen, and electrons and bonding them all together to form this water molecule. So again, oxygen is the final electron acceptor. That is important. I would know that. Hydrogen ions move from the areas of higher concentration to lower concentrations, and just like in the light-dependent reactions, they're going to move through ATP synthase. This is going to make it turn, and it's going to force that phosphate group onto ADP, making it ATP. Now, this is called oxidative phosphorylation. The substrate level phosphorylation we saw in the Krebs cycle, and the substrate level phosphorylation we saw in the glycolysis. That was a enzyme taking a phosphate group from a molecule, a substrate, and binding it to a DP, making it ATP. Now, in this type of phosphorylation, remember, phosphorylation is just adding a phosphate group onto another molecule. Specifically, in this case, we're talking about phosphorylating a DP into ATP. What happens here is we're using some type of electron transport chain, and it's going to be coupled with the oxygen molecule that I showed you as the final electron acceptor. That means that is going to be an oxidative process. So without that, the electron transport chain, this would not turn, this would not create that concentration gradient, and we would not see this type of ATP production. So this is a little bit different. It's going to use that electron transport chain to create the ATP in this case. Here's what it looks like all together. Again, we can see our NADH and FADH2 molecules donating their electrons. They're going to move through the electron transport chain. They're going to bind with oxygen at the end and form water. Now, this is also going to force those hydrogen ions into the intermembrane space. It's going to move through the ATP synthase, making it turn and creating ATP. This is what it actually looks like. I love these images. You can see it turning and making the ATP. I love looking at these mitochondria. It looks so cool. Or the mitochondrial ATP synthesis, yeah. I think I already showed you this video. No, I'm not going to play it. Here we go. Now, I like to show this because I talk about the concentration gradient being high to low. What actually is happening, it's true, it's going from high to low. What's actually happening is we have a positive and negative charge that are created by those hydrogen ions. Remember that hydrogen ions are just protons that have a positive charge. So what actually happens is you're creating a positive and negative charge, and these positives are going to want to move to the negatives, and that's actually the true reason why there is a movement of hydrogen ions or protons, is they want to equal out these charges. All right, so let's talk about the reason why we get 36 ATP for around 36 to 38 ATP per glucose. For every NADH molecule, we're going to create about 3 ATP. For every FADH2 molecule, you're going to produce 2 ATP. And remember that these NADHs and FADH2s are going to come from the first 3 steps. They are going to make their way to this electron transport chain where they're going to dump off those electrons. Now, when these dump them off, it's not like 3 ATP instantly pop out. It doesn't work like that. This is more of a calculation. It's not like for, an FADH2 molecule, unbinds the hydrogen and electrons and then 2 ATP are produced. They have to do calculations on this because this is happening so quick and there's not like an easy way to observe this. But these are the numbers. Now you actually might see, I've seen some books actually have like NADH makes 2.5 and FADH2 makes 1.5. Most textbooks will have 3 and 2, but these are roundabout numbers. These are averages. Now, if you remember during glycolysis, we produce 2 NADH and we produce 2 ATP. During the prep step, we produce 2 NADH and nothing else. During the citric acid cycle or the Krebs cycle, we produce 6 NADHs, 2 FADH2s, and 2 ATP. So up until this point, we've only produced 2 ATPs. But if we do the math, how many ATPs are produced per NADH? 3. So take 10, because we add all these up, times 3 is 30. 2 times 2, because 2 ATP are produced per FADH2. So 2 times 2 is 4. Add these all up, you make around 38 ATP. Sometimes a little bit lower because, again, these 3 and 2 numbers aren't perfect, but we say between 36 and 38 ATP or on average. Yeah, this is where this is the day when the mitochondria goes from being the powerhouse of the cell to the ATP synthesis by oxidative phosphorylation. This is that day. So I don't want you to remember the powerhouse of the cell anymore. You have to learn as this is where ATP is produced through oxidative phosphorylation. All right, we're going to get into the next section here, which is 8.6. So anaerobic respiration, if oxygen isn't present, what's going to happen is the electrons aren't going to be able to unbind with the electron transport chain, which means they're going to get stuck. And the NADH and FADH, two molecules, then won't be able to drop off their electrons. So what's going to happen is you have this bucket brigade. I always like to talk about this is what a bucket brigade is. If one person stops taking the buckets, it's going to back everything up. If oxygen doesn't take on the electrons and form water, it's going to back up everything else. And you can't, this person's going to stop filling buckets because they can't get more buckets because this person's going to hold one in their hand, they're going to hold one in their hand, they're going to hold one in their hand because they can't pass those buckets on. Think of these as like the electron carriers and the electrons. Once this stops, when there's no oxygen, you're going to back everything up. So what's going to happen is the prep step, the Krebs cycle, and the electron transport chain will stop working. If you look here, again, this process is not going to work because oxygen is not present. These electrons are going to get backed up. NADHs and FADH2s won't be able to drop off their electrons. I like to relate this to taxis. Think about if the taxis can't drop off their passengers to wherever they're going. If all the taxis start filling up, no one's going to be able to get to where they're going because no taxi will be available because they will be all full. So what's going to happen is your body is going to go through something called fermentation, or bodies, I should say, because we have different types of fermentation. This is what an anaerobic process where ATP will still be produced, but it's produced without oxygen. Now, what happens in fermentation is glycolysis will react as normal, or as it does. But what's going to happen is it's going to change NADH back into NAD. This is going to create a byproduct, and we're going to talk about the two types of fermentation, but this frees up more NAD to keep this glycolysis going. Now, it sounds kind of really inefficient, and it really is. It's only producing 2 ATP because if you look here, we're just making the 2 ATP in glycolysis. But the thought here is, not the thought, the evolutionary advantage is, it's better to make a little bit of ATP than no ATP. If you're not making any ATP and this step doesn't occur, well, it's going to be a bad day. You need ATP to live. If you don't have ATP, you're literally going to die. But yeah, this NADH goes and it's going to reduce pyruvate into lactate, and it's going to free up more NAD to keep this process going until you get to enough oxygen in your body. This happens when you're going through like strenuous activity, when you don't have enough oxygen, when you're breathing hard. This is called lactic acid fermentation. Now, you create this byproduct called lactate or lactic acid, and this is what kind of builds up in your muscles and gets kind of it's hard to break down, and it's part of the reason why you feel sore the next day after a long run or a long workout. Now, this also occurs in bacteria and different yeasts. But what happens in the alcoholic fermentation is, again, you produce only a little bit of ATP. You're going to produce ethanol or alcohol as a byproduct. This is how your beers and wines are produced. It's a very similar process, but again, the end product here is ethanol, not lactic acid, and it's only doing this because it needs to free up more NAD for this glycolysis to occur. Now, if you've ever seen one of these cauldrons, maybe your parents make beer, maybe you went to like a winery with your parents, or somewhere and you saw a big cauldron like this. This is what they're doing to create that with those beers and wines. Basically, what they're doing is they're making an anaerobic chamber, so the bacteria are going to eat the sugars that they put in there, and they're going to go through an anaerobic respiration to create that alcohol, the beers and the wines. All right, this is the end of 8.6. The last section of this chapter is going to be on the regulation of cellular respiration. And I like to make the analogy between a TP and an ice cream sandwich on a hot day. I know it sounds weird, but what basically happens is if you don't eat that sandwich very quickly on a hot day, it's going to melt. We all know that. What happens, though, is ATP is going to hydrolyze very quickly into ADP, so we need to use it very quickly. You can't just store a bunch of ATP and just say, hey, whenever I need it in the future, I'll produce it. No, it can only last for a short time, so we have to produce it whenever we need it. So your body, I should say, through millions of years of evolution, we've evolved ways to regulate how much ATP is produced. Let me go back. So what happens is the cells, when they don't need ATP, they're going to stop producing ATP. And this is because ATP will inhibit earlier steps in this process. Specifically, ATP will inhibit earlier enzymatic processes. If you remember that chapter 6, we talk about those inhibitors. Yeah, the ATP will actually inhibit some of the enzymes that are working in here when it builds up. So when we have high ATP levels and low ADP levels, it's going to stop the process of cellular respiration. Again, why keep on making ATP if we have enough ATP? I make the analogy, if we have, if the lunch ladies make 500 cheeseburgers for lunch, why should they keep on making cheeseburgers? It's not efficient. I know I'm bringing that word back, efficient. It's not efficient. Why keep making cheeseburgers? So what's going to happen is ATP builds up and it's going to stop this process. However, on the flip side, whenever ATP is low, that's going to initiate this process. Whenever you have low ATP, your body's going to say, hey, I don't have enough ATP. I need to make more ATP. And it's going to initiate cellular respiration, say, hey, make more ATP for me. Specifically, I like to look at this molecule here. You can see pyruvate dehydrogenase. This is the enzyme that changes pyruvate into acetyl-CoA. The inhibitors, ATP, NADH, and acetyl-CoAs, these are all products of this reaction. Because if we have a lot of this product, why do you need this enzyme to keep on working? The activators, things that would activate this enzyme, ADP, pyruvate, and calcium. And if you think about calcium, there's a lot of calcium used when you use your muscles. So what's going to happen is this calcium is going to activate this this enzyme to react and say, hey, make me more ATP. This is all about efficiency, inhibiting or activating this molecule when you need it. I like showing, let me go back. Here we go. I like to show this image because it shows you, again, activators and inhibitors for each of these different enzymes. And you can see, again, ATP will inhibit a lot of these enzymes and ADP will activate a lot of these enzymes, regulating how much these enzymes work. Now, I like to talk about the mitochondrial DNA a little bit. Here we talk about genetics in the next couple units here. But one interesting thing about mitochondrial DNA is that you actually inherit all of your mitochondrial DNA from your mom. This is not something I talk about in regular biology. This is kind of just a very small point of genetic inheritance that is very just interesting. So you get all of your mitochondria from your mother because the egg is what has all the mitochondria in it. You don't get any mitochondria from your father. So you will inherit 100% of your mitochondrial DNA from your mother. Now, there was one case, one family where they did, but again, if you find that online, it's an exception to the rule, but every other study we've seen, you inherit your DNA from your mother. So we can actually trace this lineage back to through history, and we can look at the mitochondrial DNA inheritance. Now, what can happen is here again, you can see the sperm is going to fuse with the egg. Once it fuses, it doesn't donate any of the mitochondria. You get all your mitochondria from your mother. So what happens is, if there are mutations in this mitochondria, what we can see is that these mutations are going to be significant because if you get them from your mother, we know that they may have had some type of mitochondrial disease, but sometimes it lays not to say dormant, but it depends on what mitochondria you get. So you can see all these mitochondria that are in your mother's cells. If you get this batch, you'll have this mitochondrial disease. If you have this batch, you might not have this disease. And it's just kind of random sometimes if your mother has these mitochondrial mutations. And these are very detrimental because if you don't have working mitochondria, your chance of surviving is very, very minimal. And it's because, again, the mitochondria is the powerhouse of the cell. It produces ATP. It makes the energy that we need, or it transfers the energy that we need. If it doesn't work, it's like not having power plants. If all the power plants in the United States shut off, bad things are going to happen. I mean, we don't have electricity power everything. So this is a very detrimental disease. And I just kind of touch upon this because it talks about just inheritance a little bit. But yeah, this is the end of the chapter. All right, 8.7, we're looking at triglycerides and how triglycerides are used in this cellular process. So we talked about how glucose is used. What we're going to see is this triglyceride is made of a glycerol molecule. This is it can be converted into a G3P molecule, and these fatty acid tails, which can be converted into acetyl-CoAs. So when you burn fat, let's say you don't have enough glucose in your body, you ate, but then you're doing a really long workout and you don't have enough glucose in your body to sustain itself for cellular respiration. What your body's going to do is it's going to switch to breaking down fat molecules, these triglycerides, which again are made up of glycerol and fatty acids. These will be broken down again into G3P and acetyl-CoAs. And if you remember, these two molecules, you can see the glycerol and fatty acids, will go into the G3P will go into glycolysis and power this process, and the fatty acids will go into the acetyl-CoAs, which go into, or made to acetyl-CoAs, which go into the Krebs cycle. So this is how your triglycerides will fuel your cellular processes. Is they are like intermediates. They create these intermediates that go into this process. Now, the same can be done with proteins. You can see these amino acids can get converted into pyruvates and other substances within the Krebs cycle. So this is why you eat why you can get energy from proteins and triglycerides, not just glucose. Excess amounts of carbohydrates will then be formed into fat. The extra acetyl-CoAs can form these fatty acids, and I can go back here and show you what that looks like here. So you can see here, if you have too much glucose, it can go back into make glycogen to store for later. Same thing with the excess molecules in glycolysis and the acetyl-CoAs, they can be formed into triglycerides. So this is kind of a way that your body regulates how much food and how much fat you store and how much food you eat and intake and what you do with it, because it needs to either store it for later for whenever you don't have enough to eat, or use it for when you don't have enough to eat, just in general. Now, glucose is a very versatile molecule. I like to show this image here and say, hey, glucose has a lot of different end pathways. We have glycolysis. We have like every single one of these pathways. I'm not going to go into them, but you can see how many pathways glucose is a part of. And I actually don't even know half these pathways, but I just want to show you that glucose is a very versatile molecule with all the different pathways it can go into.